Sleep is a significant factor in both enabling and accelerating the learning of language. This includes the learning of new words and the consolidation of learned words. A critical factor in novel word learning is the understanding of a word's meaning, also known as its semantic representation. Previous research has shown that providing semantic information could enhance associative word learning, through which people can associate newly acquired vocabulary with its meaning. However, according to researchers Shimke et al., there is a lack of research examining the effects of semantic information on sleep-mediated associative word learning. Their study seeks to address this gap. In their research, Shimke et al. Extend on the complementary learning systems framework. The CLS framework outlines two systems of the human brain involved in acquiring and consolidating new words: the hippocampal system for initial rapid acquisition and the neocortical system for slower integration into our stored vocabulary. The effectiveness of sleep-based consolidation of new language is enhanced by slow-wave sleep and spindle density during sleep, both of which are important cognitive functions for memory consolidation. This study's overall aim was to establish the impact of sleep on associative novel word learning and to determine whether the addition of semantics alters learning performance. More specifically, it aimed to investigate the impacts of sleep on recall when novel words are paired with either semantic attributes, such as related adjectives, or uncommon proper names. In order to test this aim, the researchers measured recall of novel words in a group that experienced overnight sleep compared to a control group who did not sleep. The authors hypothesized that A. Sleep would improve cued recall accuracy, and B. The addition of semantic context to the words would further help cued recall accuracy.
The study was approved by the Human Research Ethics Community at the University of Queensland, and consent was granted by participants. Participants were healthy adults aged 18 to 30 who did not present knowledge or diagnosis of conditions impacting learning or sleep and had normal or corrected to normal vision and hearing. Participants were required to speak English as their predominant language and were not to have completed overnight shift work in the month prior to the study. Measures Novel Word Learning Task. The novel word learning task consisted of 20 images of aliens, each paired with a 3-word name. The name started with a novel word followed by 2 adjectives in the semantic condition or 2 proper names in the proper name condition. Sleep and alertness measures. Participants were required to wear a sleep estimating device, the Actigraph GT3X plus accelerometer, on their wrist for 24 hours. To estimate sleep quality, participants completed the Pittsburgh Sleep Quality Index and the Morningness and Eveningness questionnaire. To measure activity, participants self-recorded their daily activity, exercise, sleep, and caffeine consumption. Finally, the psychomotor vigilance task was used to measure participant vigilance, and the Stanford Sleepiness Scale was used to estimate sleepiness. Cognitive measures. A reduced and modified version of the attention network task was used to measure attentional performance. Participants had to process spatial cues and select a direction of the most central out of 5 arrows while ignoring distracting stimuli. Attention was measured through reaction time. The reading span task. Participants were asked to recall letters presented one by one in the correct order, then read sentences and judge whether they made sense. Finally, they were given a series of sentences, each followed by a letter. Afterwards, they were asked to recall each of these letters that had followed the sentences in order. Procedure Session 1. Before the session, the Stanford Sleepiness Scale and Psychomotor Vigilance tasks were completed. The PVT involved waiting for a red dot to appear on a screen and clicking the mouse as quickly as possible. This was followed by a different word learning task. There were 5 blocks of on-screen learning during the learning phase. There was a 500 millisecond break between each trial. Each trial began with a fixation point, a cross for participants to look at. An alien was then shown with its 3-word name below. Alien word pairings were followed by a blank screen. The process would then repeat. Participants were shown the image of 20 aliens with their word names below. In each block, each alien and name pairing was presented twice. Cued recall. After the learning period, participants completed the immediate phase of the cued recall task. Participants were shown the image of the alien and required to type the first name or novel word. Following cued recall, participants had a 12-hour break before the next testing session. The break period was 12 daytime hours for the daytime condition and 12 hours of overnight sleep for the sleep condition. Session 2, the delayed session. First, there was a repetition of the Stanford Sleepiness Scale and Psychomotor Vigilance task. There was then a repetition of the cued recall task with randomized order of alien images, followed by the reading span task.
In looking at the results for this experiment, the cued recall accuracy exhibited by the sleep and the wake groups was similar during the initial learning session. As we can see on the graph, trials 1 to 5, which represent the initial learning sessions, show no difference between the sleep and the wake groups, as indicated by the colored lines. Any differences in the abilities between the sleep and the wake groups to learn and recall the novel word-object associations was negligible. The delayed testing session, however, is when differences started to appear, which we can see visually represented by the gap between the lines at trial 6. We can see that the sleep group demonstrated a significantly higher cued recall accuracy score than the wake group, with the sleep group scoring at around 1.2 and the wake group's scores around 1.0. This shows that overnight sleep does make a difference in helping in the process of novel word association consolidation, and therefore memory retention. Additionally, with the blue line indicating the sleep group who weren't given semantic information, and the green line indicating the sleep group who were given semantic context, we see that semantic information did not affect language learning in any significant manner. This is because both the semantic sleep group and the non-semantic sleep group scored at 1.2. So both groups did better with memory recall after the overnight sleep, regardless of whether they were given semantic context of the words or not. There was no difference in their performance. This proves the first part of the hypothesis for this study correct, that sleep benefits language learning, and negates the second, that semantic context further benefits cued recall accuracy. When we look at the sleep measures, they included the PSYC score, the MEQ score, and the TST score measured by actigraphy. Once again, the sleep and the wake groups had no notable differences in terms of any of these measures. For example, we can see on the graph here that the PSYC global score for both groups was around the 5-point mark. So this means that we can rule out the sleep measures as being an influencing factor of the cued recall accuracy performance of either group. We can also apply this to alertness, as the findings from the PVT between the two groups at either session also showed no significant difference. An aspect in which the groups did exhibit difference, however, were the cognitive measures. The wake group evidenced greater working memory capacity as measured by their higher scores in the reading span task. Contrastingly, the sleep group performed better in the orienting network of the AND. This can be seen in the highlighted scores on the graph over here. However, these differences are cognitive and the results don't show them to correlate with the memory retention being tested. Ultimately, there are no significant correlations between the differences in cued recall scores and any of the cognitive measures, sleepiness tests, or total sleep time for either group. So that is, none of these factors influence the cued recall accuracy of participants. Consequently, the study finds that overnight sleep significantly improves the memory retention of novel word-object associations compared to daytime wakefulness. Additionally, the effects of sleep on word learning are not modulated by the provision of semantic information.
A strength of the study is the use of both objective measures such as cued recall accuracy and actigraphy, as well as the subjective measures of participants' self-report to assess word learning, sleep quality, and attentional capacity. This allows for a more comprehensive understanding of the variables involved in the study. Another strength is the contribution it has to existing data, emphasizing its reliability. It also adds to existing literature where the findings contribute to the understanding of how sleep influences the retention and recall of novel word-object associations. However, there are limitations to the study. There is a limited scope in the experiment as there are other influential variables such as individual cognitive abilities, motivation, and instructional techniques that can play a role in the work learning which was not discussed in the experiment. There is also a delay period where the study examined sleep and wakefulness on word retention after a 12-hour delay. This provides insights into short-term memory consolidation but did not capture the potential long-term effects or changes in retention over extended periods. Furthermore, there is an interference in the wakefulness condition where participants who are in the wakefulness condition were more likely to be exposed to more waking interference from their daily activities compared to those in the sleep condition. The interference could have potentially impacted word retention and introduced confounding variables. There was also a limitation in the neural monitoring used in the experiment. The study could have utilized more direct measures for neural activity, like techniques such as polysomnography or functional magnetic resonance imaging. These direct measures could have also provided more detailed insights into the neural mechanisms underlying sleep meditated word learning.
The present article found that having a period of sleep between learning and testing led to better novel word associative learning. This research is useful because it demonstrates how sleep can help associative learning early on. However, the research shows only that the stage of sleep itself can help associative word learning. The daytime group eventually goes to sleep anyway, so they might have higher performance after sleep. We don't know if it's the sleep itself or the time of learning that is beneficial. Tamminen and Gaskell found that it takes 7 days before the strongest association between novel and similar definition words emerge. Consolidation may occur across a longer time frame or in many stages, one of which is sleep. Walker et al. In 2019 found that going to sleep immediately after learning did not improve integration of novel words. Instead, there was some suggestion that studying a longer time before sleeping led to better integration or learning. Wang et al. Found that learning new words at 8 p.m. compared to 8 a.m. led to better retention after 24 hours. Together, these studies show that sleep itself is beneficial in associative learning to help consolidate novel words and link them to other words or pictures. However, it's not yet possible to pinpoint exactly how long before sleep is the best time to introduce novel words, nor how this affects longer-term retention after 7 days, which is when the strongest association should emerge. The aim of this experiment is to identify whether introducing novel words a certain number or range of hours before sleep will lead to better associative learning in both the 24-hour and 7-day terms. 100 first-year undergraduates of an equal gender split will be selected, preferably during a non-exam or assignment-intensive week. All participants must be available for 30 minutes every day, any time between 8 p.m. and 10 p.m. for the 7-day duration. Firstly, participants will be asked for their regular or most common sleeping time. Like in Shimp et al., all participants will be asked to wear a sleep tracker on their wrist for 7 days during this experiment. This will record when they actually sleep and for how long. Then, all participants will receive a remote rendition of Shimp et al.'s novel word association test at 8 p.m. each day. Participants may begin at any time before 10 p.m., but their actual finish time will be recorded. All novel words will be introduced with an alien picture and 2 semantically similar words, such as happy and glad. There will be 20 novel words introduced across 5 learning blocks. In each learning block, the order of the novel words will be randomized. 2 testing blocks will follow. The first block will be similar to Shimp et al.'s, where 20 aliens are randomly introduced and participants asked to name the novel word associated with it. The second testing block will display each of the 20 novel words in a randomized order. Participants will be asked to type the semantic meanings. Either of the 2 synonyms introduced with the novel word or synonyms of those words under a thesaurus will be accepted. The sleep monitors will determine when the participants have actually fallen asleep, and a measurement of how long this was after they finished the exercise will be recorded. 7 days later, participants will be recalled to the lab at 8 p.m. The same tests undertaken by Shimp et al. Will be done to ensure that over the 7-day period, no moderator variable, such as sleepiness or attention change, which might alternatively explain the results, has emerged. Participants will then complete the same 2 testing blocks. In block 1, an alien picture is displayed, and the participant is asked to type in the associated novel word. This tests picture-associated learning. In block 2, novel words are displayed, and participants are asked to type in the associated meaning of that word. This tests semantic associated learning, albeit in a different way to Shimp et al. Data will be analyzed to explore whether there is any correlation between the timing of learning before sleep and associative word learning outcomes. This will help us identify whether there is an optimal time and the number of hours before sleep to introduce novel words for associative learning.